All right, so start off this lecture, we're going to talk about this bell ringer really quick. What is the difference between an endergonic reaction and an exergonic reaction? If you remember from last time when we talked about these terms, an endergonic reaction is going to be one that requires an input of energy, whereas an exergonic reaction is going to release energy going from reactants to products. So that's just a review from last time. In this lecture, what we're going to do is talk a little bit about enzymes and dive deeply more into like the actual structure of an enzyme. We talked a little bit last time about how an enzyme reduces the activation energy, and we'll mention that again in this PowerPoint if you don't really recall what that meant. So we're going to go ahead and just jump right in. The learning objectives for this lecture are basically going to be able to describe the properties of an enzyme, explain how enzymes affect the rate of biological reactions, explain how changes to the structure of an enzyme may affect its function, and explain how the cellular environments affect enzyme activity. All right, but before we start with that, there is a great Amoeba Sisters video on enzymes you might have watched in regular bio a couple of years ago. I'm going to provide the link for that just in the description of this video if you're watching this on YouTube. So definitely check this out first if you don't remember a lot from enzymes and if you kind of struggle with just remembering the concepts of that. It's a great video talking about all the different types of stuff that we're going to mention within this lecture. I'm not going to play the video right now, obviously, because of copyright infringements and things like that. So just check it out. It's in the description. So talking about enzymes, we're going to show enzymes as different shapes as we go through this lecture, but this is a great depiction of what an enzyme might actually look like. You can see here, looking at what enzymes are actually made out of. If you remember, an enzyme is basically the final structure of a protein. It's a polymer. It's made out of chains of amino acids. And if we remember protein structure from a couple of units ago in Unit 1 when we talked about macromolecules, proteins have primary, secondary, tertiary, and sometimes quaternary structure. And you can see all of that right here in this diagram. You can see the alpha helices and the beta sheets, and how we have two subunits here kind of combining together to form this enzyme. So this is actually what enzymes would look like. But for the sake of this lecture, we're going to kind of describe enzymes as like these blobs or just very simple shapes like circles and squares and things like that. What an enzyme is, again, it's a macromolecule, specifically from proteins that can catalyze or speed up a reaction by lowering the activation energy. Okay, they are not consumed by the reaction, so enzymes are kind of like a tool. Basically, they can be reused over and over and over again, similar to how like a hammer can be used every single time, like more than one time. You don't use one hammer per one nail. It can be used over and over again. And enzymes will almost always end in ASE or ACE, and that's just the naming convention that we use for enzymes. And that's how you can also identify enzymes. Like if you are reading a prompt and the prompt mentions a certain molecule that ends in ACE, you could pretty much guess that it's going to be an enzyme. All right, so just taking a look at this diagram, this is a terrific diagram showing us the enzyme function and how it works. So again, we're going from this very complex shape of enzyme that we have here in the bottom right and going to like a more, very simple clip art shape. We have like this is supposed to represent an enzyme, and then we have like these two sites that are cutaway parts that are called active sites. And the active site is going to be the region where the enzyme will actually do its reaction. And this is the part where the substrate, or the thing that is actually going to be changed from within the enzyme, that's where it's going to bind. It's going to bind right here in the active site. And the substrate will bind to the active site, and they're held in by very weak interactions. It's basically kind of like a puzzle fit. There's no covalent bonds or anything like that. It's basically just it fits in like a peg, and it will stay there until the enzyme will kind of wrap itself around it. So at this point, the substrate is converted to the products once the enzyme has gotten a hold of the substrate. When the substrates first join in with the enzyme, they form something called an enzyme substrate complex. So we refer to that pretty often. and then very quickly the substrates will get changed into the products, and usually there's some kind of input of energy in the form of ATP. Finally, the product is released from the enzyme, and then the enzyme can go ahead and do the reaction again. So this idea of induced fit is basically when the enzyme will actually change its shape of their active site to allow the substrate to bind better. So going back to this original diagram, we did see that the substrates bound to the active sites right here. And we did mention this very weak interaction, but the enzyme will almost like hug the substrates. It'll do this thing called an induced fit just to make sure that the substrate stays in place and doesn't go anywhere during the reaction so that it can actually change that substrate into a final product. And so again, this enzyme substrate complex will look like this, and this idea of induced fit will last until that substrate turns into the desired product. Two forms of reactions that we'll see, and we did talk about this in section 1, how there's catabolic and anabolic reactions. Enzyme catabolism, this is when enzymes help break down complex molecules. So suppose we have, let's represent this arrow looking shape as maybe a disaccharide. And the enzyme will come up to this disaccharide. It will do this thing of induced fit. And then once it breaks apart that disaccharide into monosaccharides, it will then release those monosaccharides, and it's breaking down something. So the idea of catabolism is basically taking a larger molecule and breaking it down into smaller subunits. And likewise, the opposite of that is going to be enzyme anabolism. This is where enzymes help build complex molecules where we want to make something more complex. So we have two smaller subunits being combined together. Again, the idea of induced fit still exists here. So you can see that the enzyme kind of conforms around the substrates. And finally, when the final product is made, the enzyme will release it and then proceed to repeat the reaction again. These two graphs are really simple when we first take a glance at them, but they're very important to understand how enzymes work and how the concentration of enzymes will work. So I'll show you another graph in just a second, but that'll make a little bit more sense. But these two graphs are really important to understand when we take a look at the X and Y axes. First, let's take a look at graph A on the left, where we have a substrate concentration on the X-axis, and on the Y-axis we have the reaction rate. So no clear units, but we know that as we increase the substrate concentration, the reaction rate will increase as well until you get to a specific point, and then it'll kind of level off. And the reason for this is because there's a specific number of enzymes that exist within a cell. And so at a certain point, increasing the amount of work available to do doesn't mean you're going to output more product. So think of it like an analogy of like maybe people working on an assembly line. Just because you have more materials available for the assembly line to work doesn't mean you're going to output more product unless you have more workers to convert the materials into a product. So this graph shows how eventually there will be a maximum, they call it a maximum velocity of the enzyme's ability to do work, just because there's a specific number of enzymes that exist. In graph B on the right side, we see a linear relationship between the reaction rate and now enzyme concentration. So going with that same analogy as the assembly line, if we increase the number of workers, then of course we're going to be able to increase the amount of product that we can create from the materials that are provided. So and that relationship is going to be linear. For every one enzyme that we increase by, we can see a linear relationship and a linear increase of the reaction rate. All right. I found another graph off of the web. I apologize for it being kind of crooked here, but this is just the way it came from the internet. This is another kind of like representation showing us the actual enzymes, showing us this graph on the left, graph A. Just by increasing substrate concentration doesn't mean that we'll be able to output more product, because the availability of enzymes gets capped to a certain point. So there is a maximum velocity at which the enzymes can work at, and it's because they're at full capacity when it comes to like how much they can work. They are fully saturated, like that point says. Okay, so let's talk about the efficiency of enzymes. How can we improve the efficiency of these enzymes? If we're considering these enzymes to be kind of like the workers of the cell, kind of like the workers of an assembly line, how can we improve the efficiency, or what are some ways that we can decrease efficiency, or some things that we need to be careful of so that we don't decrease the efficiency? Well, it can be affected by these three major things. We've talked about these things briefly before. Temperature and pH are definitely ways that we can change the efficiency of enzymes. But also we can do it with chemicals. Remember that a change in shape means a change in function. So any of these three things can change the shape of the enzyme. So what we're going to be doing with these three things is basically just changing the final shape, maybe taking it from, those folded alpha helices and beta sheets, and then we unfold them with one of these three things, and that'll eventually change the function of the enzyme. Now, there are optimal conditions in which enzymes will want to work in, like a specific temperature, a specific pH, and that will allow the enzymes to function optimally and at their very best. The rate of the enzyme activity increases with temperature due to just, laws of thermodynamics, and increasing in temperature means that there's an increase in kinetic energy. But that will only happen up to a certain point. After that certain point, the kinetic energy will then transfer to thermal energy, and that thermal energy will shake the molecules up enough where it'll actually start to break the bonds within the enzyme, and the enzyme will denature. That's a vocab word I hope you remember from regular bio. But what that means is basically you're going from that, let's say, a tertiary structure in the enzyme. You're breaking all those bonds, and you're going to be left with only the primary structure. Okay? Enzymes also work at a specific pH. So very, very important to understand that the pH ranges cannot change very much, because being outside of that normal pH can cause hydrogen bonds in the enzymes to break. Remember, pH is just a measurement of available hydrogen ions, and if you increase the number or decrease the number of hydrogen ions, you can definitely change the shape of an enzyme. And it really doesn't take that much of a big pH change in order to upset a lot of these enzymes. I'm not talking about jumping from, like, let's say, a pH of 7 to a pH of 8. We're talking more of like changing the pH from 7.4 to 7.3. You might remember from chemistry that the pH scale is logarithmic. So the change from a 7 to an 8, let's say, is like a 10 times difference, but a 7 to a 9 would be like 100 times difference. It wouldn't be 20. It's not a linear scale, it's logarithmic. So we have to keep that in mind, that the more we increase the pH or decrease the pH, the more the enzyme is going to change. This graph is just a very basic demonstration of how specific enzymes have optimal temperatures. Not all enzymes are created the same. Not all enzymes have the same optimal conditions in which they will want to work at. There are enzymes, for instance, in your stomach that will function just fine in the harsh acidic environment of your stomach at a pH of 2. Other enzymes will not. So it's really important to understand that. This graph is showing a difference in temperature, however, how there are enzymes that will work just fine in 20 degrees Celsius. There will be enzymes that will work their best at 55 degrees Celsius, which is pretty warm. It's like maybe 120 degrees Fahrenheit or so. And then there's more ones that are really good at operating at close to 85 degrees Celsius, which is extremely hot. It's not quite boiling, but we're getting close to boiling. So there are enzymes that will work at really, really hot temperatures. And it's really important to understand that not all enzymes are the same, and that each enzyme has a different optimal temperature and pH range that it will want to be at. One other thing we want to talk about with enzymes is sometimes they have these things that they attach themselves to help themselves function. And these things are called cofactors. Cofactors are not protein. They are not made out of proteins. They will assist with the enzyme function. They are usually made of something that's inorganic, so nothing carbon based. They're going to be inorganic cofactors, usually consisting of some type of metal. They can be bound pretty loosely to the enzyme, or they can be bound pretty tightly so that the enzyme has a good hold on it. And another name for that is something called a holoenzyme. That's going to be an enzyme with a cofactor attached to it. A great example of something like this that I can give you is something like hemoglobin. We've always heard that there's iron in our blood. Where exactly is that iron? It's attached to these enzymes, four of them specifically called hemoglobin, and there's one iron molecule attached to those four enzymes. There are also things called coenzymes. They're going to be very similar to cofactors where they help assist the enzyme function, but they are actually going to be organic based. Things like vitamins are really important to these things. And that's why a lot of times we have to take our vitamins or we need to eat foods that have a lot of vitamins in them to assist these enzymes to do their functions. Now, we talked about how we can help these enzymes with cofactors and coenzymes, but we also want to talk about inhibition. And so enzyme inhibitors are basically ways that we can reduce the activity of specific enzymes. Inhibition can be permanent or it can be reversible. The way we can make permanent inhibition for enzymes is by binding to the enzyme with a covalent bond. Remember that covalent bonds are pretty strong, pretty hard to break up. Examples of this would be like a toxin or a poison. If you're bitten by something that has venom, or you eat something poisonous and it starts inhibiting a lot of your enzymes, that's usually a permanent thing, and it's pretty hard to clear that out. It usually results in a lot of cell death. Or sometimes if you have like an antitoxin or an antivenom that can counteract that, it will stop the covalent bonding of the toxin. There is such thing as like reversible enzyme inhibition, and this is going to be something with a very weak interaction, usually some type of medication that can definitely be reversible over time. And we're going to talk about different ways we can inhibit. There are several different ways, the first of which is going to be something called a competitive inhibition. Competitive inhibitors reduce enzyme activity by blocking substrates from binding to the active site. So again, this active site is going to be the region where the enzyme is going to be doing its activity. This is where the substrate wants to bind to the enzyme, and that's where the enzyme will do the induced fit and break that substrate up or combine it with something else. And so competitive inhibition can be reversed with increased substrate concentration. So for example, in this diagram, we've got this teal enzyme, and then the substrate's like this goldfish looking thing. But suppose we have this competitive inhibitor that is going to go into the active site, preventing this goldfish substrate to entering the active site. The way we can reverse this is just by simply increasing the concentration of the substrates. Not to say that we are completely getting rid of all the competitive inhibitors, but the more supplies you have here, the more substrates you have, the better luck the enzyme will have to bind to a substrate versus a competitive inhibitor. And like I mentioned, this is like a reversible thing. Usually that competitive inhibitor will exit the enzyme, and then at some point, the substrate will be able to bind into this active site here. Another form of inhibition is something called non-competitive inhibition. And this is where the inhibitor will bind to an area other than the active site. Another term for that is something called an allosteric site. And that is actually going to change the shape of the enzyme. And when you change the shape of the enzyme, you also change the function of the enzyme. So in the diagram, I think it's just best to take a look at this diagram. We've got the same teal looking enzyme with the goldfish substrate trying to get into this active site. And this is a normal interaction. And sometimes there will be another site, like over here, there's a cutaway for this enzyme to fit something like a square shape. If something were to come in here and bind to that allosteric site, then that would change the conformation of the active site, and the substrate would not be able to bind with the active site. Note that the allosteric site is not a site that will actually do a reaction. No reaction is actually taking place here at the allosteric site. It is simply just a regulatory site for the enzyme so that we can control whether a substrate is being turned into a product or not. A lot of times this is completely intentional. You might think that this is an unintentional thing, but there are instances in which we have feedback loops within an organism that the product will actually act as a non-competitive inhibitor. And we'll talk about that in just a second, and it'll make a little bit more sense then. But just to add a little bit more about allosteric sites, there are two binding sites to enzymes usually. There's an allosteric site and there's an active site. And like I mentioned before, the active site is the site that is doing the reaction. The allosteric site, that's just a regulatory site, and it's not doing any reactions, and it's completely separate from the active site. Now, we can use that allosteric site to regulate. We can use the non-competitive inhibition that we just talked about and actually use it as a form of regulation. And this is when molecules bind to an allosteric site, which changes the shape and function of the active site. Now, this may result in inhibition, but also, in rare instances, we can also use it as a form of stimulation by an activator of the enzyme. So this is really an interesting thing, and I think it's going to make more sense when I show you the diagrams. But suppose we want to activate an enzyme. What we can have is something called an allosteric activator. So suppose this enzyme on the left is our entire enzyme, and the active site are these little openings here, which are currently closed off. You can see that they are completely closed off. And then we have this one molecule here that will come in and bind perfectly within the little gap here of the enzyme, and that is going to be binding to the allosteric site. When that happens, the active sites then become open, and now the cell is able to, the enzyme is able to use that active site to perform any reactions. An allosteric inhibitor is going to be doing the exact opposite of that. So suppose we have this enzyme that normally has active sites on it, and those active sites are by default open. Sometimes an allosteric inhibitor could come in, go into that active site, the allosteric site, and now the active sites are closed off. And now when that happens, the active site can no longer perform its function, and the enzyme becomes inactive. There's also this concept called cooperativity, and this is where the substrate binds to one active site on an enzyme with more than one active site, and that will stabilize the active form. So it's considered to be a form of allosteric regulation since binding at one site changes the shape at other sites. So suppose we have a substrate and it finds its way into one of these active sites. So suppose this active site was actually supposed to be open instead of closed. When a substrate comes in and binds to one of these active sites, it will actually open up the other active sites as well. So it's kind of a rare thing to see. We won't talk about it too much, but it does happen where a substrate binding to one active site will open up additional active sites on the enzyme. Now, this is what I was trying to talk about when we first mentioned the non-competitive inhibition. I did want to talk a little bit about this feedback inhibition, about how this can be completely intentional for the enzyme to have an allosteric site that blocks its ability to do its function. So when the end product of a metabolic pathway acts as an inhibitor to an early enzyme in the same pathway, this is called feedback inhibition. A really important topic to understand. And this is only going to happen when you want to have a fine balance between substrates and products. So suppose your substrate here is this green triangle, and you have intermediates, and your final product is going to be this yellow circle. And suppose you want to have a very fine balance between the amount of substrates you have and the amount of products you have. Let's say you want to have like a 50-50 mixture at all times throughout the cell's life. So what you'll have is an enzyme with this allosteric site here. And that allosteric site will actually fit the product shape here. So the enzyme, we'll start over here. Enzyme number 1 will take this green triangle substrate. It'll turn into some type of intermediate. Enzyme 2 is then involved, and enzyme 2 will form the final product. That final product will, as we increase the concentration of this, it is going to be more and more likely that the product will find its way back to that original enzyme. And when this enzyme binds with the product, that allosteric site on enzyme 1 changes the active site, and then this enzyme will no longer be able to function, and it'll kind of slow down the reactions. And so then you'll slowly build up your concentration of substrates again. And the pathway continues once the product is used up by the cell. So once all the product is used up and the substrate concentration is high, then you can repeat this whole process where enzyme 1 will then be able to convert the substrate back to its product. But basically, again, this is a way to control, to make sure that you're not using up all your substrate to create too much product. Because if you do create too much product and you don't need it all, it's kind of a waste for the cell to do that. So in this way, if we make the allosteric site the same shape as the product and it kind of fits there, then the product itself can be the regulatory thing to make sure that the enzyme's not making too much of it. All right, and that concludes our discussion on enzymes. There's a lot of different topics we talked about, but none of it's really super complicated. So I definitely encourage you to check out some of the vocab on Unit 3 with enzymes. And there's an additional enzyme worksheet that I would like for you to take a look at. You'll definitely need to know how enzymes work for pretty much the rest of the year to kind of understand their functions and the ideas of inhibition and activation and things like that. In the next upcoming notes, we'll be talking about photosynthesis and then finally cellular respiration at the very end.